Friends, now we are going to study the life cycle of Anthoceros, which is a bryophytic member commonly called as hornwort. Systematic position or classification of this Anthoceros, it belongs to kingdom Plantae because it is autotrophic and stationary one. Division Bryophyta, then class Anthocerote, order Anthocerotales, family Anthocerotaceae, and genus Anthoceros, which is commonly known as hornwort. Then external morphology of gametophytic thallus. Here the gametophytic plant body of this Anthoceros is a thalloid, dorsiventrally flattened, green or dark green in color, and in most of the species it is prostrately growing, whereas in a few species it is erectly growing. This is a Anthoceros himalayensis, which is having prostrate thallus. Then this is a Anthoceros crispulus, and this is a Anthoceros gemmulosus. These are the dorsiventrally flattened thalli, whereas this Anthoceros erectus is having somewhat erect thalloid gametophytic thallus. Now, this gametophytic thallus is a bilobed or pinnately branched or spongy with large number of subspherical spongy bodies like shimmy, or raised on a thick vertical stalk-like structure that we can observe in some of the species of Anthoceros. Now, this gametophytic thallus that has a dorsiventral symmetry is showing certain variations on dorsal and ventral surfaces. The dorsal surface of thallus may be smooth in some of the species, or may be velvety or rough with the spines and ridges in some other species of Anthoceros. But the characteristic feature of the dorsal surface is that it is having shining and thick in the middle, and this shining thick at the middle gametophytic thallus is without distinct midrib. If you observe here, in almost all species that have sketched here are without distinct midrib, which is a characteristic feature of the liverworts that belongs to class Hepataceae. And here in case of Anthoceros, you can't observe such a distinct midrib. Then ventral surface bears many unicellular smooth-walled rhizoids, and these rhizoids, which are unicellular smooth-walled one, they are present in the region of middle portion or midrib region, which is not distinct over the dorsal surface, but these rhizoids, they are present on the ventral surface. These rhizoids that are unicellular one, and all these rhizoids are smooth-walled. In an earlier member like liverwort such as Rixia and Mercansia, you might have studied the rhizoids are of two different types. One that is smooth-walled and second one is a tuberculate. But here all the rhizoids, they are smooth-walled. Then these rhizoids perform a basic function like a root. That is, they fix this entire thallus firmly to the substratum, and secondly, these rhizoids absorbs water and minerals from the soil in which they are anchored or they are attached. The tuberculated rhizoids, I have told you that they are totally absent, and scales are also absent. Instead of these, you can have such a spherical or sometimes irregular dark spots which are opaque one on the ventral surface, and these are nothing but these structures, they are commonly called as a mucilage cavities, and these mucilage cavities, they are schizogenous in origin. That means the content degenerate and it results into formation of a cavity. That cavity remains opened, and again, as it is on the ventral side, some of the blue renal members like nostoc inflate the cavity, and therefore such mucilage cavities remain filled by means of nostoc colony. And it is said that this nostoc is living there in a symbiotic association. So this is about the external morphology. The next point is a internal structure of gametophytic thallus. Here, this is a vertical transverse section of the thallus of Anthoceros in an outline. If you observe this, it appears to be more or less boat-shaped. In the middle region, it is highly thick, and as one passes or goes towards the margins, it is tapering one or very thin at the margins. Then another characteristic feature of this vertical transverse section, here you can have a uniform organization. That means it lacks the zones that we have observed in a Mercansia, where in case of Mercansia there are two distinct zones. On the dorsal surface there is a assimilatory or photosynthetic zone, and on the ventral surface there is a storage zone. But such a zonation is totally absent in case of Anthoceros, and therefore this entire thallus is uniformly composed of thin-walled parenchymatous cell, and the thickness of the middle region varies from species to species. It may be 6 to 40 cells in thickness. The minimum 6 cells thickness is observed in a Anthoceros levis, whereas maximum 30 to 40 cell thickness in this middle portion is observed in a Anthoceros crispulus. Here, the outermost, if you observe this thallus under high magnification, the outermost layer is upper epidermis, and the epidermal cells are regularly arranged. In a earlier bryophytic plant that we have studied, there is a presence of air pores and air chambers. So here, as the epidermal cells, they are arranged compactly without any gap or any space. Here epidermis is a defined one and not having any opening or air pores. So the epidermal cells, they are regularly arranged, smaller in size, and have a large lens-shaped chloroplast. So each epidermal cell, as well as all the parenchymatous cells, they are having such a large lens-shaped chloroplast. Each cell of thallus contains either a discoidal or oval-shaped chloroplast. The shape of the chloroplast is a discoid or oval, and each such chloroplast encloses a single large conspicuous body called as a pyrenoid. So here another characteristic feature of the chloroplast with its definite shape is a presence of pyrenoid, and this is a characteristic feature of class Anthocerote. Presence of pyrenoid is a characteristic feature of Anthocerote. Now here all these cells of this thallus, they are performing function of photosynthesis, and at the same time the food material is also stored. So each and every cell performs both the function of photosynthesis as well as storage. Now, some of the cells of this ventral surface, some of the cells from the ventral surface are characteristic. Here, in a intercellular cavities are present on the lower surface or ventral surface, and these cavities are formed due to the breakdown of the cells. That means they are cystogenous, and the cavities remains filled with mucilage, and therefore they are called as mucilage cavities. But these cavities open on the ventral surface through a stoma-like slit or pores called as a slime pore. So if you observe, this is a mucilage cavity formed due to the cystogenous activity, and it remains filled by means of a mucilage, and it is open on the ventral side or lower surface of the gametophytic thallus. Now this cavity is covered by means of a pore which is commonly called as a slime pore. Now this slime pore remains open forever, that cannot be closed like stomata. So you will get confused the pore as a stomata, but this is not a stomata, it is a simple opening, and it is therefore called as a slime pore. Now this slime pore, as remains open with the ventral surface, usually the mucilage dries, and at the same time some of the nostoc species enters into this cavity, and usually they grow there in this mucilage cavity. And the nostoc that grows in this mucilage cavity, which here they have shown, is usually regarded as a symbiotic association. That means this nostoc and Anthoceros, they are growing together in a close association in such a way that there is a give and take mechanism, and therefore commonly it is called as a symbiotic type of association. So here the next point is a reproduction in Anthoceros. Now this hornwort Anthoceros reproduces by vegetative and sexual methods. Vegetative method of reproduction takes place by means of death and decay of older portion, by tubers, then the next is by gymi, by persistent growing apices, and fifth one is by apospory. So we have to study one by one. By death and decay of older portion of the thallus, which is commonly called as fragmentation, which is very common in most of the bryophytic members. So this method is not common in Anthoceros, which is very common in case of liverworts. Liverworts मध्ये हे फ्रॅगमेंटेशन मेकॅनिझम जे आहे, फ्रॅगमेंटेशन मेथड जी आहे ती more common आहे. It is not so common in case of Anthoceros. The older portion of the thallus usually disintegrates, and due to the aging or drought, this disintegration takes place, and due to the disintegration of the basal portion of the gametophytic thallus, usually the branches get separated, and each detached lobe or branch later on develops into a new independent plant by means of apical growth. Then second method of vegetative reproduction is by tubers. These tubers are perinating structures produced during unfavorable conditions or prolonged drought. The marginal tissues of the thallus get thickened under such circumstances, and it results in the formation of tubers. Their position varies in a different species. They may develop behind the growing point, as in case of Anthoceros levis, or along the margins of the thallus in case of Anthoceros hallei or Anthoceros pearsoni. In a case of Anthoceros himalayensis, the tubers are stalked and develop along the margins of the ventral surface of the thallus. So these tubers, which are perinating structures rather than reproductive structures, develop at different sites on a ventral surface, either along the margins or along the midrib portion or along the tips of the growing thallus. Now each such tuber has outer 2 to 3 layers of corky hyaline cells, which encloses the tissue containing oil globules, starch grains and aluminum granules. So these are the storage products, and they are capable to pass on the unfavorable conditions. As they are developed during the unfavorable conditions or prolonged droughts, entire thallus get degenerates or dry, wilt and destroyed. But due to this outer 2 to 3 layered corky hyaline cells, this tuber remains in a dormant condition, and on return of the favorable conditions, tubers produce new thalli of the Anthoceros. Then third one is by Jimmy. In some of the species of Anthoceros, many multicellular stalked structures develop along the margins of the dorsal surface of the thallus, and these structures are called gymni. When detached from the parent thallus, each gymna develops into new plant. Now this presence of gymni is a characteristic feature of liverworts like Mercansia, but such gymni are present in some species of Anthoceros, which is again not a common method of vegetative reproduction in case of Anthoceros. Then fourth is by persistent growing apices. Now what happens in some species of Anthoceros, due to prolonged dry summer or towards the end of growing season, as these bryophytic members, they are shade and moisture loving, usually growing on damp, shady situations. But when the conditions become dry, the conditions become unfavorable, the whole thallus dries and gets destroyed except the growing point. The apical growing point remains as it is, and this apical growing point in some species grows deep in the soil and becomes thick under unfavorable conditions, and this underground portion develops into a new thallus on approach of favorable condition. Thus, throughout the dormant conditions or throughout the unfavorable condition, it remains as a dormant, and on approach of the favorable condition, it germinates to develop into new plant. Thus, it is a perination rather than multiplication. Then, fifth type of vegetative reproduction is by apospory. In a Anthoceros, unspecialized cells of many parts of the sporophyte, for example, the intercalary meristematic zone cell or subepidermal cell and sporogenous region of the capsule, form the gametophytic thallus. Now, this phenomenon is called as apospory because all these cells, they are developed from the diploid zygote after mitotic division. So each and every cell of this group is a diploid, and this diploid cell directly develops into gametophytic thallus, which is usually haploid. But under such circumstances, when the phenomenon of apospory is there, the gametophytic thallus develop from the sporophytic cells or a sporophytic tissue or a sporophytic region other than the spores is called as apospory, and this thallus, gametophytic thallus, is always diploid, but even though it is a normal in appearance, and such phenomenon of apospory is more common in Anthoceros levis. So this is about the vegetative reproduction in case of Anthoceros. We are going to study the sexual reproduction and structure of sporophyte, that is the remaining part of the life cycle, in a next lecture. Thank you.